Yay. Welcome. You guys probably all know her, but I am so excited to introduce Dr. Andrea Berardi, who has been an HUH fellow and working in the lab for a couple of years. And before that, she got her PhD at the University of Virginia studying Silene, then went off to learn a ton of amazing skills in biochemistry, molecular evolution, and phylogenetics, and working with Stacy Smith at Colorado, and then Chris Coulimar on petunias. And we are all very excited to congratulate her on her new position as a faculty member at James Madison University. So we are crying that she will be leaving us, but so thrilled that she gets to share her wisdom with a whole new community and start her lab. And so now we'll hear what's she's been up to and what she'll start off doing.
Hey, thank you, Robin. Hey, hello. It's been a while since I've seen a lot of you. So I'm really happy to just tell you about what I did with my fellowship at the Herbarium. And it is basically this: characterizing red floral color evolution in polyploids. And we're going to go light on the polyploids today because they're really tricky. Okay, so my favorite thing is to look at color, and color varies across all kingdoms and all systems. You know, it's in fungi, it's in animals and plants and all sorts of things. So we all know and love color. But what's really fun is that plants, in particular the angiosperms, have really harnessed this trait, and it's very, very diverse across, especially floral tissues, not always floral tissues, but really diverse across the whole group of plants. And in addition to being variable across these large macroscopic scales, we also know that floral color can vary within a genus and even within a species, sometimes within an individual. So it's a fascinating trait for me. So overall, the broad question that I focused on over the past 10 or so years, maybe more, who's counting, is how does variation arise across different scales? And so, like I said, I've focused across the years that I've been studying floral color across different levels of variation, including within population variation, among population variation, local adaptation, reproductive isolation and speciation, and now more on the macro scale with lineage diversification. And sort of these are the three sort of categories that I've been, or stories rather, that I've focused on. But today I'm just going to talk to you about this one, which has been funded by the Harvard University Herbaria. So the main question that I focused on while I've been here is, how does red floral color play a role in population, speciation, and lineage diversification in Silene? So this is a Silene, and this, there we go. It just is Robin, sorry. My mentor for this project is Robin Hopkins. So the reason why you haven't always seen me here is because, you know, I spent some of my time here, and then also out at the Arboretum, which is where the Hopkins lab is based. All right, so why red flowers? Red flowers occur across the angiosperms, but it's often associated with hummingbird pollination. So often when we think about red flowers, you often think about hummingbirds. Not always the case, but there's a very strong association. Red is interesting because it can arise through several different genetic mechanisms. So one way that red can arise is it can be a gain from white or no color. So that's an example of this would be this red petunia here. It can be a shift from a different color. One great example is flax. And another way that we can think about how red arises that may not be a super obvious way is actually through the act of genome duplication, or polyploidy. And when you get polyploidy, this can induce something called transgressive phenotypes, or in this case, pigmentation. And what that looks like, for example, in the terms of red floral color, is you can have, say, a white parent and a pink parent. This is Nicotiana. And then suddenly you get a red hybrid offspring. So that's when the phenotype is so different from either one of the parents, transgressive. So another way that you can get red that maybe gets a little less attention. Okay, so red is also really uncommon in the angiosperms. It's sort of counterintuitive because if we think about our gardens, we see red flowers all the time, but that's because humans love red. In fact, when we look phylogenetically, red is very, very rare. And that's, for a couple different reasons. The first is that hummingbirds are often the main driver of red, and hummingbirds have a restricted range. Hummingbirds are only in the Americas. The other thing is that biochemically, red is a challenging color choice, I will say. And that's just because when you get red, you often have to break different parts of this pathway that all plants have, and that could be deleterious. Another thing is that red is sometimes associated with lower diversification rates. So a couple different reasons why we think red is rare. So what is red? First, let's take a step back and let's actually quantify what red is. Why do I have to do this? This is because people, color is subjective. So I'm going to give you an example of Silene and challenges that I've had looking through literature as well as through herbaria records, also just talking to people. These three species are often categorized as red. So this one is called the red campion, and to me, this is not red, this is pink. But a lot of people are very passionate about this being red. This one is kind of in the middle. It's called the rose campion. People often tag it as red in databases, but I'm not sure that it's actually red. And then we get this one, which I would call red, but it's called the Californian pink, which is unhelpful. So as an aside, the pink, I think, actually refers to the pattern of the petals, referring to like the pinking patterns such as pinking shears, but that's besides the point. Okay, so given that there's a lot of different ways to define red, that can be problematic. So we need to actually find a way to categorize this scientifically. So we can do this two general ways. One is look at the biochemistry. So there are two main ways of making red floral color. The first is anthocyanins only. So anthocyanins are floral pigments in the flavonoid pathway. All plants can make them. They just sit tight in the vacuole. So that's the main method. Another way is to use anthocyanins and carotenoids, and they just lay on top of the vacuole. So these are carotenoids in the chromoplasts here. So together you can use sort of like a pink and an orange, and it sort of makes a reddish color. So you can define it biochemically, and the other way is you can define the color spectrally. So looking at spectral reflectance of floral petals, you can see that, white flowers and yellow flowers have a very different shape. Like there's no question that's different from red, but you might wonder what it, like maybe pink and red look similar. They actually don't. So you can see very similar to purple where it has this M shape, but the red colors are much more contracted. So spectrally red is actually different from pink. So we have two different ways of actually defining red. So now we're in business. Okay, so why did I pick Silene again to sort of ask these questions about red color evolution? I did my dissertation in Silene, but on this plant right here, which is not red. But what I was always really attracted to was the beautiful red color of this species right here, Silene virginica, which grows on the East Coast. And that just sort of got me thinking, what about red in Silene could be interesting? And as it turns out, Silene is a widespread genus in most temperate regions, but the red species are all in this single clade, and this clade is only in the Americas. In addition to that, there's only 9 species of red flowering Silene, which is 1% of the genus, which is a very, very infrequent color. All species are in this clade, which is polyploid. So they're not diploid. They are at least tetraploid. Some of them are even up to 12x, which is challenging. So there is one red species that's up here, but we can talk about that another time. So it's rare in this genus. They're in a single clade, and they're only in North and Central America. So that immediately sets up a question that I can start to address. So looking at where they actually grow, one of the first things I thought of is, are these in the areas, or do they grow in the areas that hummingbirds are? And the answer is yes. Very, very broadly, what I'm showing you here is all the Silene in North America, colored by floral color. You see that red is sort of like right here and here, and these are hummingbird distributions. And sure, they do overlap, but so that's sort of a maybe. The other thing is I talked to you about transgressive phenotypes created by polyploidy. So that is certainly a possibility here because all of these are polyploids. So 2 questions. 1, is it because of hummingbirds? 2, is it because of polyploidy? They're not mutually exclusive. So moving on, where, when, and how do these lineages arise in Silene? Okay, so what did I do with the HUH's money? I I took it and ran. No, so I was able to actually collect all of the red species in person, which was amazing. So the HUH funded 4 collection trips. So one down here where a bunch of these species are, one here, here, and here. So I was able to get 7 out of 8 in person, and the 8th species, which grows over here, I was able to have a collaborator send some seeds. So that was amazing. And just a quick little picture of like, I had a great time, but also it was just wonderful scientifically to get to see where your species actually grow. And to also, maybe do some pollinator observations while you're there. This is just to show you that the red species grow, a lot of the time in weird rock places. And like up here, for example, that's my husband, who was my unpaid field assistant, and he likes rock climbing, so that was good because he went and got that plant for me. This is my colleague Jenna, and she was able to sort of climb up. So they really like rocks, but also some of the red species are in very like lush, grassy, weedy areas. So no obvious ecological or abiotic things. But the other thing is that I was able to purchase a UV camera because something that's also very important to the phenotype is whether or not there's UV pigments. Because if you have UV pigments, you can get a slight bluish hue. And I wanted to make sure that those were either there or not. And this is just an example of collecting in the field and being able to sort of spot check, are there UV pigments or not? The answer is no. Okay, so then what I did is I got situated at the HUH. I went through the collections, measured phenotypes, did a lot of biophylogeography, and did a lot of UV camera photography. So getting that data, here are the questions and just sort of the general outline of what I'll talk to you for the rest of this talk. So I wanted to know, when in Silene did red actually emerge? How many times? Is it just, one instance, or did it happen a bunch of different times? Does ploidy actually matter in the shift to red? And whether there are co-occurring floral traits associated with red? Because if we are only having hummingbird pollination cause red, we would also expect maybe some phenotypic changes such as tube length, nectar, things like that. So are there co-occurring phenotypes? And then I wanted to know, is red color convergent? Is it the same? Does red happen every way, or the same way every time? It's always coming from a pink lineage, it's coming from a white lineage. Is it the same genetic mechanism or not? And then I'll talk to you about future directions. Okay, so the first step was to ask, when and how many times did red flowering lineages arise? And I did this with an RU student the first year I was here, Anna, who is absolutely amazing. So that's just to say, this is with the help of Anna. So what we did is we went through the herbaria. So this was a remote RU. So luckily we had all the photos that we could share with her so she could actually see the specimens. So we went through and we scored floral color using herbarium specimens, as well as counts in the flora of North America, and then also just from my collections. So this is a single gene tree that we also made together. This is just ITS, just to say like, okay, where in the genus is red and what does it look like? So what you can see here is this is the polyploid clade Physolignus that the red species are in. And you can see that red is not just in one clade. So it definitely has arisen a couple of times. So that was great. We sort of answered one question. And then the next question was sort of like, when did red arise? So something that you can do is use a tool called ancestral state reconstruction, where you have the phenotypes in your extant species here, and then you can just use statistics in a phylogeny, such as this, to estimate what the likelihood of each ancestor was, what the floral color was most So what you can see here is every one of these pies is the probability of a particular color at every one of these ancestors. And what you can see is that, it's not always so clear, but what is clear is that none of these are totally red. None of these ancestral pies are totally red. So that tells us that most likely red is a very recent trait. So you don't see significant red until sort of this area. And the fact that this is a very contracted tree tells us that this diversification happened pretty quickly as well. So red evolved within, we think, the last 2.5 million years, which on an evolutionary timescale is pretty recent. Okay, so then what I did was I wanted to make a better phylogeny, because the phylogeny I just showed you is a single gene, and that can tell one story, but many genes tell a better story, and certainly with polyploids, we need to get a better handle on what's going on. So I used a custom set of baits, and I extracted DNA from herbarium specimens here, as well as some of the fresh tissue that I collected from the field. And I assembled the data and made single gene trees, and I am working on a species level phylogeny right now. This has been incredibly challenging because of the polyploidy, because each individual has at least 4 copies of each gene. And as it turns out, polyploid phylogenetics are sort of the cutting edge of phylogenetics right now, where we're not super sure the best way to do things all the time. So right now, what I'm going to show you is sort of a summary tree. So this is basically summarizing each species. So this is the general topology of the polyploids, but there may be, once we zoom in on single genes, there might be some switches, maybe some splits, but this is generally what we're looking at. So what did I want to know from this fancier tree? I wanted to know specifically how many times red arose, and then from which type of phenotype did it arise? Is it always a pink? Is it always a white? And what we can see is that there are actually 2 polyploid clades, 2 different polyploid events. And here's the first one up here where this arrow is, and there's 1 red lineage in this first one, and then the rest of the red are in this one. So these are actually hexaploids, and these are all mixtures of 4x and 8x, and then 1 really silly 12x. And what we can also say from the previous trees, we know red is not ancestral. So we know red is more recent. So again, we're sort of thinking, okay, so this red must have happened very, very close to the tips. So now we can look at those tips and sort of estimate, is it coming from a white? Is it coming from a pink? And I count up to 8 independent transitions to red. And this is sort of the summary. So looking at the likelihood of each ancestral clade, and then also what are the sister species, I see 6 transitions from pink to red and 2 from white to red. And just to note that these 2 species right here, subciliata and regia, are sister to each other. So that's a single event. And that came from a white. Most everyone comes from a pink. So that's interesting. We have at least 2 different ways of making red. You're coming either from a white or a pink. And I don't want you to focus on the actual data here, but I just wanted to show you that we also did some flow cytometry and to try to get to the question, like, does polyploidy matter? And what you can see is that there's a wide range of genome size here, and red is sort of represented at all of these different genome sizes. So in a very general sense, I don't think polyploidy matters for red, because you have red at all of these different ploidy levels. So more on that at a later point in time, but polyploidy does not seem to be the most obvious answer as to why there's red. Okay, so then what floral traits associate with red color? If red is arising from hummingbird pollination, we would expect morphological changes that all of the reds look very similar to each other. Okay, so to do this, we again used herbarium measurements on the flowers, and we also used species ranges, the trait ranges from the flora of North America. And again, my RU Anna helped me with this, as well as 2 absolutely wonderful Harvard undergraduates, Dorina and Sabrina, and also Robin. So what we did is we focused on these particular floral traits that represent just floral morphology. Okay, so again, getting at this question, do we expect color sort of to equal morphology. So if hummingbirds are the reason why red species arose, again, we would expect them to kind of look similar. And we would expect these to also look different from white species if they are, as we might expect, moth pollinated, and also different from pink species, which we expect would be bee pollinated. So if we sort of map all these traits on a graph, we might expect something like this, where these different colors are exist in different spots of the graph or the morphospace. The next question is, if we overlay where these species are in sympatry, where you have two species growing in the same area, we might expect, let's say, a red and a pink species to be really phenotypically different, because you'd think that the species would be selected to have different morphologies to attract very different pollinators to prevent hybridization. It's just a general expectation we have. So that's a question. When we look at all of these different species, if they're in sympatry, do they have really different phenotypes, or do they have very similar phenotypes? Okay, so here's the data. What we did is we took all of those traits, we phylogenetically corrected them, and it does not look like that example I showed you where we had like, red up here and pink and white. They're all jumbled together. And I think this is really interesting because we know that these species are new, and we think that this was rapidly, sort of just a rapid adaptation. And certainly the phenotypes are all jumbled up. So just to sort of show you, the whites and the pinks are all over this morphospace. And, we have really sort of long and big flowers. We have short and stubby ones. We have different shaped short and stubby ones. But pinks and whites are just interchangeable in this area. However, the red ones are just in this one area of this morphospace. And they look like this. So instead of diverging from the greater Silene morphospace, they're sort of converging on a very small part. So it could be that hummingbirds selected for these species and they just sort of landed here. You can see these all have sort of the same shape of tube, and that is certainly reminiscent of a beak, but it could also make room for a long proboscis. So some indication, but not like a slam dunk yet. Okay, so I just told you all of this, which is great. The important thing is that we actually need pollinator observations to confirm this. We can talk about what we think about pollinators all the time, but we actually need some good pollination biology. And there's only been pollination biology on 3 species in this entire North American clade. So moving forward, that's what I'd actually like to do. And now, since I will have students, I can have them do that as well. So that's going to be great and a lot of fun. Okay, and to get at this sort of sympatry allopatry question, so again, if we think about 2 species in sympatry growing together, we would expect them to be more different than each other or more divergent. And we actually see not that big of a difference, and also more of a difference in allopatry. So what this means is that species that do not grow in the same places are possibly more phenotypically different than in sympatry. So basically the opposite of what we expected. This is a very weird result, and it could be that most of these species actually can't hybridize, so maybe it doesn't matter. But a future direction of this is to actually look at a more finer scale. So this is just sort of looking at broad ranges and just physical overlap using maps. So what I'd like to do is actually look at 2 species that can breed and do hybridize, and we can look at them sort of in areas of sympatry where there's range overlap and when there's not range overlap. And we can actually try to get at this question a little more specifically. So future direction. Okay, so now, is red a convergent signal in Silene? So specifically, do red flowering Silene reflect light in the same way, kind of like what I showed you before? And then do they use the same pigments to produce red color? So if they use the same pigments, that is an indication that they might be the same genetic mechanism. So let's take a look. Okay, so here's the floral reflectance spectra. And what you can see is here are all the red species, and they all have a very, very, very similar shape. Pretty similar. When we compare it to the pinks and the whites, it's a really different shape, right? So the pink ones are all sort of this very M shape, and the whites and the yellows are just this sort of flat shape. And the reds are this, I don't know what to call it, S, I'm not sure. But I think, what we can draw from this is, yeah, they have, despite whatever chemistry they're using, they're sending the same signal, especially to what pollinators can see. So that's interesting. So what about the chemistry? So I told you briefly about the two different types of pigments that you can use to make a red color. Roughly speaking, are category 1, carotenoids are category 2, and you can have a combination. So what do we actually see? So I extracted pigments from all of these species. I also did sister pink and white, but I'm just going to show you the red today. And I'm just going to show you presence absence. So I'm not going to put a biochemical pathway up here. What I'm just showing you is if there's a plus, I saw this pigment. If there's nothing, I did not see the pigment. Sometimes I'll have 2 pluses, meaning I saw a lot of that pigment. Okay, so for example, this species, Silene serpentinicola, I found 2 different types of anthocyanins, pelargonidin and cyanidin. In this species, Silene laciniata, I found pelargonidin, which is an anthocyanin, and a carotenoid. So those are the 2 different strategies already. So, okay, we have at least 2 different genetic mechanisms, probably. So looking at the rest of the red species, it's kind of all over the place. These red species are just making red however they see fit, which is really fun. So that means this is going to keep me busy for a long time trying to figure out the different genetic mechanisms. So that seems a little overwhelming at first, but actually, if we take a phylogenetic approach, we can look at the sister species to each of the red species. And here's an example. So this is Silene serpentinicola, which I just showed you. And actually, its sister species, Silene hookeri, has the same pigments as the red species. The difference is that the red species has a lot more cyanidin. So that's already just narrowing down what genes we can target to sort of probe into this question. These are the 2 East Coast species that I will be working on more specifically in the next couple of years. And this is sort of telling the same story, but with a different set of pigments again. So again, you see these 2, there's the red, there's the pink. The red species and the pink species both make their colors with cyanidin and peonidin, but the red species has a lot more cyanidin. So again, more cyanidin in both stories, but different sets of pigments. So again, this is going to still keep me very busy, but looking at it this way is just a little bit easier thinking, okay, there's probably only a handful of genes, if not 1, that can make just more cyanidin. And so looking at this pattern, this actually gives us, okay, 1 biochemical pathway. This gives us actual clues as to which genes are the likely causes of the shift to red. So in this blue pair, it's, it could be either or both of these 2 genes, and this orange pair, this gene is probably a target. And in addition to that, could also be a transcription factor. Okay, so what are my conclusions from my time here at the herbaria? Red flower color in Silene evolved multiple times in 2 different polyploid clades by using 2 different pigment classes, both anthocyanins and carotenoids. They do exhibit a convergent long floral tube morphology and a little bit of nectar. I didn't show you the data, but they make a lot of nectar compared to their pink and white sister species. And the pigments suggest very specific candidate genes to examine the genetic mechanism of the shift to red. And that is something I'm working on now. And as Robin said, I am now gainfully employed for the next, well, hopefully forever, but I'm moving to James Madison University and I'm going to start my lab, which is really great. And I'm going to be taking the Silene show on the road. So if you know any students who are looking for a master's project or somebody interested in a postdoc, please let me know. But I have some future directions that I just like to share with you. And also if you have feedback, great, that would be wonderful. So what am I going to do at JMU? So I still have so many questions about red floral color and how it contributed to Silene speciation. So basically I've sort of gathered the appropriate information to ask very specific questions now. So question number one, is red color evolution in Silene actually due to hummingbird selection? So what I'd really like to do is do pollinator observations in the field and actually put arrays out. So with the genetic skills that I learned in my previous postdoc and also ones that I've been honing here with flocks, I'd like to do arrays with natural hybrids out in the field to see, do hummingbirds preferentially visit red versus pink? What are the other pollinators coming and doing? Who's making the hybrids? Are hummingbirds actually going and making the hybrids between the red and the pink? Or is it something like, I don't know, a butterfly? The answer is it's probably a butterfly. So a lot of questions revolving around that. We'll be setting up transgenics so we can do tests in the lab because we don't want to put transgenics out in the field, and actually switching red and pink, but sort of keeping morphology the same. The second sort of main question is, have the molecular signatures of polyploidization enabled red to arise? That's a very fancy way of being like, I think we still need to get into this polyploidy thing. That's going to be really complicated, but also very exciting. And I hope to have better and more answers for you soon. Maybe the next time you see me. And this is just, this is just a description of, these are the hybrids that I think will help me answer these questions. So Silene virginica, Silene caroliniana. This is the map here. This is where JMU is, which is super conveniently in a hybrid zone. So we're going to have a lot of fun with that. And this is actually what the hybrids look like. So here's Silene virginica, the red species. Here's the pink species. They have very different growth habits, floral sizes, similar tube length. And these are the hybrids, which look a lot like this species, but just with a darker color. So I think that will be really interesting to see what kind of hybrid it is. And this is a closer view of them. So more soon. And the other thing is I'm sequencing the genome of this species, which I think will help for so many parts of my questions. And it's a big one, but not as big as, say, flocks, which is great. Okay, so then the rest of the things that I want to think about moving forward, with the genome sequencing, we can ask a lot more questions and answer more specifically. I have a lot of molecular genetics going that I didn't talk about today, but I did get transgenics up and going in Silene. So this is a knockout of one of the pink genes right here. There is a really interesting case of UV bullseyes, not in the red species, but in that hexaploid clade that grows out in California. And so these are them. So this is the UV image of these individuals. And I think that has actually led to a shift to bee fly pollination out there. So I think that would be really cool. More about co-evolving floral traits. And then there's also, in some red species, there are rare pink polymorphisms, which herbaria have helped me find. And with that, I just really want to thank the herbaria for funding this project. It enabled me to get back to the US and also start the project that I had been dreaming about since graduate school. So I just really want to thank the herbaria for the support and the opportunity to get this project up and running. I also want to thank Robin for being my mentor for this project and really just letting me run with these Silene questions and helping me through several identity crises and ultimately getting a faculty job. So thank you, Robin. And I also wanted to mention that the herbaria has been excellent with also work-life balance, because one of the reasons why you didn't see me is because I had a baby while I was here, and now she's 18 months old. So that's my daughter. And with that, I will take any questions you might have. Short talk, this is probably the shortest talk I've ever given. So if you have questions, I'm happy to answer them. Thank you.
Thank you. Fascinating. And can you go back to the phylogeny that has the that one there? Sorry, yeah. The next, yeah. That one. Have you thought like on plotting those different anthocyanins in the whole tree? Because to me that seems to be like like a path-dependent evolution, right? Like you first have to evolve cyanidin,
Mhm.
and then you can evolve redness by adding other colors or something like that, right?
Mhm.
So have you thought about like if it's like a stepwise process of some ancestral? Because I think in the first tree that you showed,
Mhm.
that you mapped the flower color, the outgroup has like the red color, right? So probably
Yes.
something already like pre-exists in those species, right?
Yeah, so the answer is yes. I have looked at that, and I'm still waiting to get a little bit more data. But actually, you landed on one of the key things, is that cyanidin, this central pigment, is the key to moving towards red in most clades. However, pelargonidin is really the reddish, truly reddish, like orange pigment that I was expecting. Cyanidin does look red. So the answer is clade wise it's like clade wise, there are very specific patterns. And I've done a couple of those where I have the full pigment information, and it does seem like it is stepwise, but it's not a super clear signal. I don't know, that's not really answering your question, but the answer is yes, I plan on doing that with the full data set. I've done it with the smaller data set I have. And a lot of people have also done that sort of analysis, and stepwise is usually the way to describe shifts to different colors. But what's a little challenging in that is adding the carotenoids, because the carotenoids are present in all of the species, and sometimes it's the adaxial side and sometimes it's the abaxial side, or abaxial, adaxial. So that is the plan, but it's going to take some thinking. Complicated answer, sorry. Yeah. Yes, the answer is yes.
And the second question,
Yeah.
well, like here, I think in the Americas, they are over stressing all the time in the red color, the hummingbirds. But red flowers have also evolved in Africa, in Europe,
They
everywhere,
have.
right?
They
So
have.
what do we know in the literature of why is red evolving in those places where there are no hummingbirds, right?
Yeah. It is a really good question. And there's a lot of different answers, because red is kind of, it also depends on which pigments you're making it with. So for example, like poppies, which, that's what I think of when I think of, like, let's say Eurasia. I'm like, oh, poppies. That's mostly carotenoids, actually. So number one is there's different biochemical strategies to making red across continents. The other thing is there's a lot of different abiotic factors that red can be advantageous for or not. So I think there's a lot of different abiotic reasons, as well as selective reasons, that could still be pollinator related. Butterflies think red is great. They just don't necessarily always go to red. They like a lot of other colors. But it's an active area of research for sure. So I don't have actually a great answer for you, other than like there are definitely red flowers outside of the Americas. But I think it will matter on which biochemical pathway they're using. And then also what abiotic factors, temperature and moistness seem to be pretty important. So, yeah. Yeah.
Hi, thanks, Andrea.
Yeah.
Congratulations on the job.
Thank you.
I also have a few questions.
Great.
So you're taking spectral reflectance, right?
Mhm.
And yet you're also measuring the pigments in the lab. Are people using spectra independently to just estimate pigment concentration specifically for these?
Yes, you can.
And that work, do you understand it to work really well, or?
It works okay. I think, like, is it going to necessarily pick up which anthocyanin is there? Probably not, because what my data shows is even though you're using different pigments, you're getting that same signal, that same spectral shape. So you might be able to tell the difference between anthocyanins and carotenoids, because carotenoids have a slightly different spectral absorption pattern and reflection pattern. So in a general sense, you could say, is this overall signal red or pink? And you can sort of use it as a proxy for amount. That's totally kosher. But which pigment is a little bit more difficult.
Yeah, that's interesting, because I only work with leaf reflectance, and the spectra that you showed, my interpretation of those, even just within reds, which were certainly more consistent than pink and cream.
Mhm.
But even just among the reds, I thought there was a lot of variation.
Yeah.
And anyway, my general take with spectra is that it's within leaves is that you can do a great job of distinguishing a lot of functional traits,
Mhm.
characteristics.
Mhm.
So,
Yeah, it's not as clear cut with floral pigments. The other thing that I didn't tell you, and that could be leading to the variation your keen eye saw, was that some of the red species do have UV pigments, even though generally red flowers don't. There was some within population variation, and that can certainly change the UV or the whole spectral shape in the UV area. So there's that, and also, maybe some of these different pigments are actually leading to those slight shifts. I wouldn't put a lot of money on being able to say which pigment from which spectra, but yeah, I think for me, they looked very similar in that compared to pink or red, or pink or white, but yes, more to do for sure. But there's a lot more of the chemistry that I chose not to go into today.
and second, would you tell us sort of where you're at with the polyploid phylogenomics? Just a
Sure.
little bit more about
Love to.
what you're doing.
Okay, so the summary tree that I showed you, I have all of the haplotypes per individual. I have those phased, and now I'm very confident in what those are. I don't think there are any chimera. So I think I have all of the different haplotypes per gene. So one strategy I took to make that tree was to use astral, which just, again, sort of makes a species tree. And that very cleanly groups each species and all of its haplotypes with itself, which might suggest autopolyploids. But there's other evidence when we look at the single gene trees that there might be allopolyploidy. So with the general species tree, I'm confident that the astral tree shows like this general relationship, certainly between chunks of clades. There might be some more recent hybridizations within clades. And for that, what I'm doing is I'm working with Patrick, because he knows a lot more about this than I do, is we're using phylo-networks and looking for actual like hybridization, which could represent allopolyploidization, just using those haplotypes. And that I'm doing like clade-wise. And then what I'm doing is coupling all of that together and trying to figure out if there are any deeper allopolyploidy events. The biggest struggle is that there are no extant diploids in this clade, which is challenging because there's nothing to compare it to other than more distant relatives. So it's hard to sort of polarize whether these allopolyploids are like, oh, that's like these haplotypes are from this parent and these are from that parent. So that's where I am. So I have I think I'm very confident in the sort of the groupings, but then within each grouping, I'm still working on the finer aspects of the potential allopolyploidy. That's how I'd summarize it.
Being confident in the groupings, that's what you need.
For my questions right now, yes. Yeah, it's gotten me to a point where I'm like, okay, we can ask some macroevolutionary questions. Honestly, doing ancestral state reconstruction on trees that demonstrate hybridization that's not really a capability at this point, at least my capability. And there's a couple very recent papers that can do that. But if you're aware of anything, let me know. Okay. Yeah. Thanks. Yeah. Jen.
So I love the pictures of all the Silene up in rocks and then kind of
Yes.
in these really beautifully messy areas.
Yes.
But that made me wonder, are the rocks like a certain type of rock? Are we getting more calcareous? Is it more calcareous soil or something? Is there some other maybe like habitat specific geochemistry that's going on that I've now expressed everything I know about habitat geochemistry, just so you know?
Yeah.
But
Yeah.
I'm just, that was my first thought about that.
Yeah, it's a really good question and something that I have also been wondering. And I'm hoping to get an interested student who perhaps knows a little bit more about geology than I do. So yeah, some of them do grow on those rocks. My cursory knowledge is that they are not all the same type of rock. And the ones on the West Coast are actually more like red. And the ones on the East Coast are more limestone based. But that's again, the extent to my knowledge. But I think that would be really interesting for sure. I'm collaborating with an undergrad student at Christopher Newport University, and he's actually looking at some of the abiotic aspects using the bioclim data set. And it does seem like the red species compared to sister pink and sister white species inhabit warmer climates and wetter climates. But it really depends on a smaller scale as well. So I'm definitely interested in that, and I think that is an excellent avenue to pursue. Yeah, are the rocks the same? Are they not? Is it just that it's a rock? Yeah. Yeah, Patrick.
Thanks. That's a great talk. I'm curious about the hybrid zones. Have you seen them in person, and what are they like? Like, are you seeing like, I don't know, fully hybrid populations or just like one-off hybrids? And like, do you expect there to be enough hybridization in nature that you can use those hybrids alone in nature to get at kind of the genomic basis for the red color change, or not?
Yeah, I love the question. Okay, so I'm familiar with this hybrid zone here because I did my PhD in Virginia and down here. So these are the ones that I saw. I have not personally been to this area to look for plants, that is. And mapping them, this was news to me and this was news to me. So I would love to visit these other sites and look around, but this I do know. So the potential hybrids are in two places in Virginia. One is sort of right outside of Charlottesville, and the other one is a historical population in Blacksburg, which is where Virginia Tech is. Sad news for the Virginia Tech population. It is now a subdivision. So those are gone, but there are herbaria specimens at the Virginia Tech herbarium. So if I wanted to look back, there's some things I could get from that. But the area around Charlottesville, along the Ravenna River, there are a couple of persistent populations. And these species tend to be, they're called like short-lived perennials. So am I sure that the plants I'm looking at are the same ones I looked at 10 years ago? I don't know, but it's the same population. So there are persistent populations that we could return to and monitor. A lot of them are really dwindling though. So there's that. So there's a lot we could do with those that we find in nature. And then also you can actually cross these two species, which I have F1s out at the Arboretum growing really slowly. So I'm planning on doing sort of like a two-pronged approach, like one artificial crosses and two observing in nature. And it would be great if I could find more and vigorous populations elsewhere. But yeah, so they do persist along this river. Along the river, there's also a lot of the red species, but I don't see any of this species yet. They look more like that species. So a lot of cool questions. Does that answer your question?
Yeah, I think so. That northern population looks like there are a lot of purple dots there.
Yeah,
Do you expect
yeah.
that to be like a bigger hybrid zone, or you just don't know yet?
I don't know yet. Geographically, it looks like it should be, but I don't know. I think Pitt has a field station in this area that maybe I could go hang out at. This is Pyman tuning or something. Yeah, this certainly I'd bank more on this than these right now. So yeah. Kanchi.
Within Silene, where are the nectary glands located? Are they inside the tube?
Yes, they're right at the base.
Situation with the color pattern in hibiscus. You may remember that in the fall semester, someone from UK gave a seminar, and she did the genome analysis on the different color pattern, whether there is any similarity between hibiscus and silene.
Mhm.
That's one. And another thing that dawned on me is before we are culturing the poinsettia plant, wherein the leaves themselves, not the flowers.
Mhm.
So it's so much variable in color pattern from red to something else. Whether whatever you have been talking, whether can be applicable to the leaf color.
Okay, number one question was, where are the nectaries and are they similar to the hibiscus?
In silene.
In
I know
Silene?
that.
Okay, yeah, okay, in Silene, there's actually, it's not very variable across species. They are all at the base, at the anthophore, basically. They're just all there, that doesn't change. And it's just the amount of nectar produced is the variable trait. The second question,
So now, whether there is anything that is comparable between hibiscus color pattern and silene color pattern.
gotcha. There's a lot of color variability across multiple tissues, including in Silene. And I think actually you've hit upon a point that is really important in the evolution to red, is that most plants are able to produce cyanidin in the vegetative tissues. So that's one of the reasons why totally shifting to a pelargonidin approach is actually dangerous in a way, because if you can't make cyanidin, that's generally the first anthocyanin that's made in the stress response vegetatively as well. So how I said earlier that cyanidin seems to be the key to being able to choose which color you are, it also reflects the sort of the importance of maintaining that part of the pathway. But I don't know if I have a solid answer to your question other than that. Like, yes, cyanidin is important. There are a lot of vegetative patterns. Like you can even see here that different parts are also reddening. But I can't see any patterns as that pertain to my questions with the vegetative tissue. I did do a lot of that for my dissertation. And I think actually the more interesting questions about the vegetative patterns in Silene at least are within species versus among species. Does that answer your question? Yeah, okay. All right, I think we should get out of here so the other people can get in. Thank you so much for your attention.